Glycogen phosphorylase, an enzyme long associated with the breakdown of glycogen in muscle and liver tissues, is now garnering attention as a potential therapeutic target in the fight against neurodegenerative diseases. Its primary function, to catalyze the phosphorylases of glycogen into glucose, 1-phosphate, makes it a key regulator of cellular energy availability. But what was once considered a peripheral player in brain metabolism is now being reevaluated as researchers uncover its significant presence and activity within astrocytes and possibly even neurons. This shift in understanding opens up promising avenues for targeting brain-specific glycogen metabolism to combat disorders like Alzheimer's disease, Parkinson's disease, and other forms of progressive cognitive decline. In the healthy brain, glycogen stores are relatively limited compared to those in muscle or liver, but they are strategically located within astrocytes, the star-shaped glial cells that support neurons both structurally and metabolically. During periods of high neuronal demand, such as learning, sleep-wake transitions, or recovery from oxidative stress, astrocytic glycogen can be rapidly mobilized through the action of glycogen phosphorylase. The resulting glucose derivatives are then shuttled to neurons, helping to maintain synaptic transmission and buffer against metabolic stress. This astrocyte neuron lactate shuttle plays a crucial role in brain energy homeostasis, particularly when systemic glucose supply is interrupted or when neuronal mitochondria are under duress. In neurodegenerative diseases, however, this finely tuned metabolic support system begins to unravel. Impaired glycogenolysis can result in local energy deficits, increased oxidative stress, and the buildup of harmful protein aggregates. At the same time, an inability to clear or regulate glycogen may contribute to the formation of polyglucosan bodies, insoluble glycogen-like structures that are often observed in aging brains and in specific diseases like Lafora disease. These pathological accumulations can disrupt intracellular architecture, impair signaling, and create a toxic environment that accelerates neuronal death. Enhancing the activity of glycogen phosphorylase in this context could help clear these harmful deposits while also improving real-time glucose availability to stress neurons. Activating this enzyme might also provide another crucial benefit: improved redox balance. Efficient glycogenolysis via glycogen phosphorylase not only supplies fuel for ADP production but may reduce reliance on anaerobic glycolysis, thereby decreasing lactate accumulation and reactive oxygen species (ROS) formation. Since oxidative stress is a central hallmark of most neurodegenerative diseases, including Alzheimer's and Parkinson's, maintaining a healthier metabolic profile in vulnerable brain regions could help preserve mitochondrial integrity and limit further neuronal damage. Additionally, by supporting cellular energy needs more effectively, activated glycogen phosphorylase might help prevent the chronic low-grade inflammation that often accompanies neurodegeneration, as energy-starved microglia and astrocytes can shift toward more inflammatory phenotypes when deprived of glucose. Importantly, glycogen phosphorylase exists in multiple isoforms: brain, PYGB, muscle, PYGM, and liver, PYGL, each encoded by a separate gene and regulated by different signaling pathways. Therapeutic strategies that selectively activate the brain isoform, PYGB, without affecting systemic glucose levels or hepatic glycogenolysis would be crucial to avoid unwanted side effects like hypoglycemia or excessive muscle catabolism. The development of brain-permeable small molecules or gene-targeted approaches to modulate PYGB activity could, therefore, represent a highly specific and low-toxicity intervention. However, caution is warranted. The brain's relationship with glycogen is delicate, and overactivation of glycogen breakdown may lead to unintended consequences. Excessive glycogenolysis could result in imbalanced lactate production, osmotic stress, or depletion of glycogen reserves needed for acute neuroprotection. In ischemic conditions or seizure states, for instance, brain glycogen is rapidly consumed as a last resort fuel, and an inability to restore those reserves could leave neurons more vulnerable during future metabolic stress. Any therapeutic that modulates glycogen phosphorylase would need to carefully balance the rate and extent of glycogen mobilization, possibly through time delivery, feedback-inhibited drug designs, or pairing with mitochondrial support agents. Nonetheless, the potential benefits are compelling. A therapeutic approach that enhances brain glucose availability, reduces oxidative stress, and simultaneously clears harmful glycogen byproducts addresses several root contributors to neurodegeneration at once. Unlike treatments that only aim to reduce symptoms or slow plaque buildup, Targeting glycogen phosphorylase could strengthen the underlying metabolic resilience of neurons and glia alike. When combined with other metabolic interventions such as exercise mimetics, ketogenic therapies, or insulin-sensitizing drugs, the activation of this enzyme may offer a powerful addition to multi-pronged treatment strategies. In the long run, understanding and manipulating brain-specific glycogen dynamics could revolutionize how we approach neurodegeneration, not just by treating symptoms or halting a single pathway, but by restoring a deeper form of cellular balance that supports long-term brain function. As researchers continue to unravel the complexities of astrocyte neuron metabolic cooperation, glycogen phosphorylase stands out as a uniquely druggable target with the potential to shift the paradigm from damage control to metabolic preservation.